vectors are DNA molecules that are used to transport. They are vehicles, just like a car which carries people from one place to another place, like Ola and Uber. Ola and Uber are there. So likewise, in biotechnology, there is a vector which carries people, which carries, instead of people, it carries certain genes from one place to another place. And what is that place? Here, this is host. Like what host? Like E. coli. So we'll simply clone the gene of interest in a vector and you will put it inside the host. So likewise, Ola goes from destination A to B. So here you clone your gene of interest into the host. Here destination B is your host. So without cloning vector, molecular gene cloning is totally impossible. So you'll not be able to clone any gene and you will not be able to study its functions, how it is expressed, what are the elements inside it. You will not be able to do anything. So that's why vectors are very, very important. Now I'll see what is the workflow of this vector, how this vector is used in gene cloning. So first of all, suppose there is E. coli bacterium and bacterial chromosome is there. This is plasmid. Plasmid is a self-replicating extra chromosomal DNA. It has nothing to do with the chromosome. It is extra chromosomally expressed. So bacteria, first one is there is isolated plasmid. First they have isolated the plasmid, then cut the plasmid with an enzyme. Although these plasmids are commercially available in the market, you don't have to worry about just isolating these plasmids from this bacteria. It is available in the market. Then this is your required gene of interest. So this will be gene of interest. It can be any cell. It can be any mammalian cell or some other cells. Whatever you want to study, you will just isolate the DNA. Gene of interest will be this. This will be a gene of interest. Then cut DNA with same enzyme. Suppose you cut the vector with enzyme EcoR1, EcoR1 and HIN3. Okay. So here also you will use EcoR1 and HIN3. You cannot use EcoR1 and EcoR1 and HIN3 here, and here you cannot use EcoR1 and KPN1. So this is very important concept. You must have to use same enzymes in your gene of interest and in vector. You cannot use different enzyme, because if you will use different enzymes, your sticky ends will be different, because KPN1 has different recognition site. So it will cut in another site and it will make different sticky ends. So your gene of interest will contain a different sticky ends. Suppose this is your gene of interest and this is the vector. So this is the vector. Let me take another color. Suppose this is the vector and this is your gene of interest and you want to clone it inside this vector. So this vector needs to be cut and this gene also needs to be cut. So the vector which will be cut, it will be cut with EcoR1 and HIN3, and your gene of interest will also be cut with EcoR1 and HIN3. It should not be like EcoR1 and KPN1. Why? Because it will generate different end. So your vector will not combine with your gene of interest, because KPN1 has different recognition site as compared to the HIN3. That's why we use same restriction enzyme to create the sticky ends, compatible sticky ends, so that it combines with your gene of interest. The vector combines with your gene of interest, as you can see here. These two ends are cut by same restriction enzyme, and these two ends are cut by same restriction enzyme. And at last you get the recombinant plasmid. Okay. Now you see what is the history of the restriction enzyme. So scientists Harvard Boyer and Keliachi, Itakura and Arthur Riggs worked in Boyer's Lab, University of California, and recognized a general cloning vector with unique restriction site for cloning in foreign DNA and expression of antibiotic resistance gene. Then why the antibiotic resistance gene is used? Because suppose you will clone your gene, inside this vector, and you will clone this gene inside your vector, and it will go inside a host like bacteria. It goes inside a host like bacteria. So what will happen? This bacteria will take up your plasmid containing your gene of interest. And when you will grow this bacteria, it will grow with lot of bacteria, lot of host cells which will not contain your recombinant DNA also. Recombinant DNA means your plasmid with gene of interest. And in order to select your particular recombinant DNA or particular plasmid, you must add antibiotics or some selectable marker so that only that bacteria will grow, that which bacteria will contain your gene of interest containing plasmid. That's why selectable marker is very important in case of cloning. So this is the importance of the antibiotic resistance, and this is also called selectable marker. Why it is called selectable marker? Because it is used to select the particular recombinant plasmid from a population of the different from population of a particular host like E. coli when you transform your recombinant plasmid inside the E. coli. So the vector size is here is very small, like 4 kb, and we are talking about PBR 322, which was discovered in 1977, and it had two antibiotic resistance in what it is. If you know, please write in the comment section. I will be very happy to hear from you. What is the resistance gene present inside the PBR 322? Okay, please write in the comment section. Now you see what is the history. In 1961, genetic recombination demonstrated. In 1970, isolation of restriction enzyme that selectively cut DNA. In 1979, restriction enzyme mapping of the simian virus SB40 was done. In 1974, NAB opens for business. In 1975, launch of the rebase restriction enzyme database. In 1976 to 1977, introduction of Maximum Gilbert sequencing method. I have already discussed Maximum Gilbert sequencing method in my DNA sequencing method video. If you have not watched that video, kindly go and watch that video. You will have a very good understanding of Maximum Gilbert method. 1977 report of the first cloning vector, that is PVR 322, that we have already discussed. And tell me about this marker. What are the selectable marker in the comment section? Now 1978 Nobel Prize awarded for Smith and Albert Nathans for the discovery of the restriction enzymes. This you must remember for the exams. Some exams they ask very means memorable questions, like memory based questions. So you have to ask, you have to remember these kind of things. Now I'll see what are the features of the cloning vectors. So all commonly used cloning vector have some essential features like origin of replication, as we have already discussed. This makes a autonomous replication in the vector. Origin is a specific sequence of the nucleotide from where replication basically starts. So The